Annette von Drygalski. I'm a professor of clinical medicine here at the University of California, San Diego. And I am a hemophilia person, bleeding and clotting, but several years ago have picked up PPH for various reasons. And I want to say thank you to a lot of people, particularly Dr. Patricia Silva, who is our Mozambican colleague, who I see she's on. And also my sister Andrea is on because she heads the foundation who has in part funded this project. And after we started, Patricia, Dr. Silva in Mozambique, and I started this project in 2018, I want to say, with 1 year of preparation and run-up. And it's now 2026. And the project is finished and we have the results. And these are the first results of the finished project you are hearing today. And it's not, that data are not only in part published, so please keep it a bit confidential as you feel it's right. But I would love your input at the end of the day. There is lots to digest, and I think we're just scratching the surface of it all. So thank you, Homa, for giving me the opportunity here. So the project is called Postpartum Hemorrhage and Mortality in Sub-Saharan Africa, results from an intervention study. So the background, I know I'm talking to the choir, but still, women's health affects half of the world. And maternal mortality varies around the globe. It's very high in Sub-Saharan Africa and Southeast Asia. Just talking maternal mortality in general, not even PPH. If we think about 100,000 live births in Africa and Southeast East Asia, it's 300 to 1,000, whereas in Europe and USA, it's maybe 5 to 20. So there is a huge gap here. And PPH, that's why we are all here, is thought to be responsible for a third of maternal death worldwide. So data on contemporary incidents, contribution to mortality and rescue interventions, though, are relatively scarce. So the loss of a mother is obviously always catastrophic, but it's particularly catastrophic in these regions because the life of a whole family often depends on the woman. So, and that's tragic because PPH, as we all know, is preventable, it's treatable, it's under-recognized regarding effects on public health, particularly in this environment where it happens most often. And in addition, it's a difficult environment for reliable data collection and really poor local awareness. So the causes of PPH in many ways are those four famous T's, which is tone for uterine atony, tissue, retained placenta, trauma for lacerations and uterine rupture, and then clotting, the two T's here, the coagulopathy, which brings us all together here in hematology and OBGYN. So the objectives here were San Diego and Maputo have a long-standing relationship in global health that predates our PPH project by about 20 years, originated in infectious diseases and AIDS. And so Dr. Silva and myself were banking on that infrastructure and taking advantage of that existing infrastructure to build our own collaboration and conduct that project. So we wanted to study PPH, risk factors, and mortality in metropolitan Mozambique, and to inform then intervention strategies to reduce PPH and maternal death. The methods. This was a prospective data collection of deliveries at Maputo Central Hospital, which is the largest university hospital in the capital, Maputo. And we had a pre-phase run-up from 2019 to 21, then interrupted by this intervention installation of cryoprecipitate production in conjunction with early transfusion and standardized PPH care protocols, such as, for instance, single-use hemorrhage boxes, PPH education and simulation, as you can see here. But really, the intervention was cryoprecipitate production. And for that, we had 2 training sites, one in South Africa and one here in San Diego, Dr. Patricia Silva and her blood bank colleagues like Dr. Sandra, she is here, for instance, were then trained to deliver that technology to Maputo Central Hospital and train staff. The data extracted, you can see here for yourself. So again, this was all prospective chart collection with each delivery. And PPH determination was by charted diagnosis, by a blood loss over 500 milliliters, transfusions charted, and or notes indicating significant bleeding. And that was approved by both ethic committees on both sides. So here are the results. We have a cohort of 16,835 women divided in pre-intervention, about 8,800, and post 7,900. HIV was present in about 10% of the women, which is what is published for that area. And then about 2/3 of the women were married. The rest was single or unknown. And about half was urban versus rural. Rural meaning really outside the capital, Maputo, and urban in the capital. So about, let's say, 70 to 80% of women had either 0, 1, or 2 children before, and then there was a minority actually with 3, 4, or more birth. Birth mode was vaginal in 57% and C-section 37%, and the rest was unknown. And there was a median weeks of gestation was 39 weeks. So now we come to the money shots, the main outcomes of interest. You can see that pre-intervention maternal mortality was extremely high, 1.1%. That means 1 of 100 mothers would die. Infant mortality also extremely high, 3.44%. And PPH also relative high, 14%. Now, post-intervention, that changed quite dramatically. You can see that there was a relative risk reduction to suffer from maternal mortality by 2-fold, and PPH by 3-fold. And infant mortality went also down based on the numbers, so many women, this is significant, but it wasn't as impressive as maternal mortality and PPH. So that translated in a death rate reduction from 1,100 to 452 per 100,000 live births. That's still a far cry from being low and where we want it in the Western world, again, 5 to 20, but it is a lot better. So now what is the PPH association with maternal mortality? In univariable analysis, the relative risk was 8.35, and in multivariable analysis, adjusted, that was still 3.34. So these are all the terms by which it was adjusted, and I'm not a biostatistician, so our biostatistical team, we have a whole team on hand at UCSD helping with that, obviously. But PPH, in the presence of PPH, maternal mortality is 3-fold increased when adjusting for all these other factors. So this is a busy table and we will not go through with it. But then the next question is, well, what are the risk factors associated with PPH? And the risk factors are residing outside of Maputo, so in the rural areas, high and low parity, preterm delivery, hypertensive disorders such as eclampsia or preeclampsia, and infant mortality. And I don't think that's a surprise. I think that fits very well with known data. But there were protective effects, and that was post-intervention, as we already know. Not only mortality, but also PPH itself decreased quite a bit post-intervention. So any kind of educational measures instituted, that's how I interpret it, were really helpful to bring down PPH per se. And something I cannot interpret, and the OBGYNs maybe can give some feedback, C-section. So if a C-section was performed, PPH was less. Sorry for that. Let's go back one. So now we come to PPH treatments and transfusions, because that was sort of our main interest at outset, given that whole cryoprecipitate project. And so you can see pre-intervention, 1,251 women suffered from PPH, and how were they treated, i.e. transfused. And you can see that only a third of these women, despite catastrophic bleeding, got a transfusion. Only a fourth, a quarter got red cells, and then a fresh frozen plasma and platelets. And since no cryoprecipitate or tranexamic was available pre-intervention. So post-intervention, there was a doubling of transfusion incidence, so it helped to institute all these transfusion protocols, and a third of the women received cryo. So you can see here that the transfusions increased by double from 33 to 69, and that is definitely true for red cells and also for fresh frozen plasma. Platelets stayed the same. Platelets are really a rare commodity in Sub-Saharan Africa, difficult to produce. But then a third of all the women got cryo, and some got tranexamic acid. I know it's on the essential medication of the WHO, and it does save lives, but unfortunately, it's not always available. So now, okay, we know that. So this is the same table up here, so don't pay attention. But then among the women who received transfusions, so, and we talk about the number of women depending on the type of blood product. So among those, the number of transfused units per bleeding woman decreased post-intervention. You can see that here from 2.39 as a mean of red blood cell units to 1.86. The same is true for fresh frozen plasma. It doesn't seem much from a mean of 482 to 418. I understand that, but the number of women is so large that even a small difference becomes statistically significant. Platelets also dropped, but what was interesting, the blood loss increased. So you can see blood loss increased here from a mean of 1,139 milliliters, just over a liter, to 1,276. Again, not a lot, you would think clinically, but again, the numbers of the women are large, so that makes a significance. So what are the risk factors associated with maternal mortality now? Among those women who suffered from PPH, whereby transfusion is then the variable of interest. And again, these numbers are adjusted by these parameters: place of residence, birth mode, hypertensive disorders, and infant mortality. And you can see that the post phase did not make a difference. So it didn't, so the mortality was the same for those who had PPH. So interestingly, while the mortality overall, we understand, dropped post, and while PPH dropped post, the mortality from PPH, once a woman suffered from PPH, did not, no matter if you were in the pre or the post phase. Transfusions didn't make a difference, but what made a difference was blood loss. So with increasing blood loss, there was increasing mortality, irrespective of pre or post phase. So the maternal mortality from PPH did not decrease overall post-intervention. And I want us to digest that and let it sink in, because I think it's important. Each 100 milliliter increase of blood loss increased mortality by approximately 9%. And transfusions, be it red cell plasma and or platelets, did not influence PPH mortality. However, when this parameter, transfusion, did it happen or no, in yes/no fashion, was separated in components, red cells per unit increased the adjusted odds ratio to die by 1.43%, and that was highly statistically significant. It was shocking to me. I would have not expected that. So each unit transfused increased mortality by 43%. And I do not have a good explanation for that yet. We really have to digest this because it matters, obviously. However, and contrasting this is cryoprecipitate yes/no. Administration was associated with a highly significant mortality decrease. So all the mortality decrease in essence experienced in this post-intervention phase was due to cryoprecipitate cryoprecipitate administration. And it, as you can see here, the number is quite remarkable, reduced the adjusted odd ratio by 0.06. So now what about cryo? So the transfusion protocol specified the administration of 10 bags of cryo. So the massive transfusion protocol was very assimilated to what we do here in Western countries. It's not 10 units in the OBGYN world. It's maybe 2 or 3 or 4 units. I don't quite remember. I have to go back. And with that came some fresh frozen plasma, and the transfusion protocol specified the administration of also 10 bags of cryo. We made sure that the minimum content of each cryo bag was about 250 milligrams of fibrinogen, so very much like Western standards. But I want everyone to note that other than fibrinogen, which we want to deliver here, obviously, cryo has also factor 8, von Willebrand factor 13, and fibronectin. So now at the end of the talk, let me come to anemia and thrombocytopenia. We had laboratory data available on admission for approximately 2,000 women, 12%. These were the women in distress. In Maputo, we cannot do blood draws on every woman coming through the door because of resource restrictions. So the physicians made a determination if someone was in distress, and then those women would get blood draws. And you can see that the anemia prevalence is quite high, as expected for the region, 55%. Most of it is nutritional deficiencies, such as iron deficiency. And if we go by mild, moderate, and severe criteria for these anemia, 55 and 6% of patients had really moderate and severe anemia. Moderate meaning hemoglobin between 10 and 7, and I think severe below 7. And thrombocytopenia prevalence was about 22%, and you can see that most of it is mild thrombocytopenia with moderate and severe thrombocytopenia in less of the cases. But why is that important? Because prenatal anemia and thrombocytopenia are predictors of PPH. You can see here on the x-axis are the hemoglobin levels on admission, and on the y-axis you have the probability of suffering from PPH. And there is this inverse relationship with an inflection point at a hemoglobin of 10 to suffer from PPH when the hemoglobin is lower. Particularly below that cutoff of 10, you see a real quite significant rise of likelihood to suffer from PPH. So pre-existing or prenatal anemia, mostly nutritional, will facilitate PPH for physiological reasons we also only understand poorly, unfortunately. And the same is true for thrombocytopenia, maybe less likely, maybe less surprising because platelets are our mainstay of blood coagulation. But at the inflection point of about 200, you can see that the risk rises quite significantly. And that's true for the pre and the post phase here in yellow and in blue. There is a risk of PPH with simultaneous anemia and thrombocytopenia. And maybe I go back this one slide again. You can see here in the anemia world that mild anemia between 10 and a hemoglobin of 7 is either not significant or maybe even almost protective. And that comes out in this table. So anemia severity, if there was no anemia, compared to mild anemia, you can see here with mild anemia, there was a protective effect. Mortality from PPH could be reduced to 0.36. And if thrombocytopenia was present, that was compounded a little bit to 0.86, but it didn't become statistically significant. However, when moderate or severe anemia was present, no matter the hemoglobin, just by categories, the risk was 2.16-fold increased, but it was strongly compounded if thrombocytopenia was present as well. Now you increase that risk to over 8-fold. So interestingly, mild anemia, we always call it physiological anemia. We all know that most women develop mild anemia during pregnancy, a hemoglobin of 10 to 12. We wouldn't blink an eye, right? So actually, mild anemia reduced the risk of PPH irrespective of thrombocytopenia. However, once more severe forms of anemia are present, it's bad, and they are compounded by the presence of thrombocytopenia with quite an increased risk. So conclusions, maternal mortality and PPH rates were extremely high, particularly during the pre-intervention phase. A substantial reduction in mortality and PPH was achieved following implementation of a multifaceted PPH prevention and intervention program driven by hematology through close collaboration with obstetrics, gynecology, and transfusion medicine, and centered on cryoprecipitate availability. Once PPH occurred, only cryo administration, not other blood products, was associated with a marked reduction in maternal mortality. And then cryo, it is producible from locally available fresh frozen plasma, emerged as the most critical blood component for reversing PPH and preventing mortality. And so this interdisciplinary collaborative model with shared responsibilities provides a potential blueprint for regional replication. And prenatal anemia was amongst the strongest modifiable risk factors for PPH, with risk increasing steeply with anemia severity. Thrombocytopenia compounded anemia-driven PPH risk. And then because of this anemia issue, addressing iron deficiency anemia during pregnancy, thereby reducing PPH susceptibility, represents an important future goal to further decrease maternal mortality. In other words, it is a modifiable risk factor. So that's it. Thanks for listening. And with that, I would like to acknowledge the whole Maputo team with special thanks to Dr. Patricia Silva. You see her here. Funding, it's the Stadima Foundation. It's our HRSA grant. That's our federal agency here in the United States. And my own UCSD discretionary funds, and Maputo Central Hospital, and the different services here for providing the infrastructure. And you can imagine it was quite an effort for the local team there to pull together all those data. So thank you.
Thank you so much, Annette. That was really terrific. I have a few questions, but I want to open up to the audience to make sure they chime in. And if I don't see one, I'll ask one. I guess maybe I'll give a comment about the C-section. Why it could have been, quote, protective, I wonder, in your data. I think perhaps, I don't know 100%, but if sometimes women undergo vaginal delivery and, in a large tertiary center, they may not have always the ratio of staff to the patients where everyone's being closely observed to check on the perineal lacerations or if there's excessive bleeding vaginally. And sometimes if they have vaginal delivery and they're okay, like they could be fine, and all of a sudden not so fine. And sometimes those women might be more, quote, at risk, but not that the C-section was protective. It's just they had vaginal delivery that had complications that might have been picked up a little later. So I think that could be one possibility. Then let's see if any other thoughts. I was kind of curious about, you were talking with respect to the red cells, being transfused, and could there be higher risk of mortality potentially from PPH? What do you hypothesize could that be from? Is it maybe some sort of increased viscosity, or maybe that they, what is it? Do you think that mechanism could be at play?
Yeah, I mean, we all know that red cell transfusions have their own caveats, right? That's why we are become so transfusion restrictive in other scenarios. That's why we keep even in the ICU, etc., we keep our our transfusions to a minimum, and have these low transfusion thresholds with a hemoglobin of 7, sometimes now down to 6 before we transfuse, because I think transfusions just carry their own risk. It's a blood product. We have to talk to our blood bank colleagues in Maputo to also look at safety. Now, I wouldn't expect that any viral infection issue manifested itself at the time of, but maybe there is something particularly to the red cell units in Maputo. I think we have to look again how they are produced, stored, delivered. And I don't know. This is really, because it's so acute. There's this whole host of literature out there in the non-obstetrics world, etc., how detrimental transfusions can be. And 10 or 20 years ago, we just poured red cells into people no matter what, and we have stopped that, right, for reasons. So I just wonder. It is a little interesting.
Yeah. I see Neil, you have a hand raised. Go ahead.
Yes, hello. I'm Neil from South Africa. Just on the transfusion issue, is it not just that the people who bled more were the ones who got more transfusion? So they were sicker in the first place, so they're more
It
likely
was
to die.
corrected for that. So the statisticians adjusted for that. So that's what I initially thought. I said, okay, so maybe those who bled more also got more transfusions, and that's the reason why. But doesn't seem to be so, because it was adjusted for.
Okay.
So
But
the blood
maybe they were
was
sicker.
adjusted for blood
Maybe
loss.
they were sicker in other ways. I mean, why would a person get more transfusion? Maybe because they were tachycardic, hypotensive, and so on, irrespective of the measured blood loss.
Yeah,
And the increased number of transfusion units may just be a marker of their poorer clinical condition,
yeah.
which
But on the other
um,
hand, then they also got cryo. Why was cryo so protective? So I think we can speculate, and I'm really, really very thankful for all kinds of thoughts here, because in a real publication, we will have to start speculate, because there's so many things corrected for, and then we just have to speculate, I guess. It is.
I wonder too, like, some women who come in and it relates to another comment I had with your anemia. Could it be mild anemia at increased risk, for complications, or no anemia, sorry. Some women who are preeclamptic, they get hemoconcentrated often. And so that doesn't mean their anemia is better per se, right? It's a pathological condition that's happening, and they're potentially at risk for bleeding too and clotting. We don't know exactly why. So I think just calling it anemia in itself, or it could be kind of masking a little bit of what kind of anemia, or if there's a secondary kind of underlying pathology going on. And then maybe I know from the Women 2 data too, I think Monica's on here from their group in the London School, they showed that even at really low blood loss volumes, you can have severe, morbidity from hemorrhage, I think. And so the amount of bleeding might not tip someone over to coagulopathy and things. So I don't know.
Yeah, I mean, that's definitely a real important point, particularly for that region, because if we always think of PPH and or at least you guys know much better. I'm a hematologist here, but always think about it has to be 500 or 1,000 milliliters or some such thing. But really, if you start out at a hemoglobin of 6, you cannot, I don't have that volume of 1,000 milliliter in you anymore. So a few milliliters or 200 milliliters or 300 milliliters are already 200 or 300 too many, correct? So that is, but that was not even charted, because it was defined as at least over 500 milliliters blood loss. But we don't even know, I think, what happens to these women who start out with such severe anemia. And it's not a minority. I mean, this is quite a big number because of iron deficiency, unfortunately. And I don't know that can be easily fixed either, quite frankly.
Thank you so much. Any other comments or questions from the audience? I don't see any hands, but if if folks think of any you want to put in the chat, feel free to, or if you think of it at the end. But I'm going to maybe shift gears. Thank you so much, Annette,
Thank
again.
you.
Terry, if you could kindly share your slides, and we're so excited to have you present as well. I always like to have a very mixed presentation of different topics and perspectives. And so if you could kindly introduce yourself, Terry, and thank you so much again for presenting all the way from Vietnam. So on your 9 p.m. time zone from where I am on the East Coast, at least. Okay.
Well, thank you, Homa, and thank you for giving me the opportunity to present. So this will be a little different. So I'll present to you the company. In that process, I'll introduce myself. I'm the CEO of Qui-Gon Therapeutics. We're a company that's going after acute bleeding. So we come at this a little differently in that we see postpartum hemorrhage as the entry indication to prove that we have something safe and efficacious and a drug. And then from there, we'll expand into other indications as well. So I'll spend my time introducing to you exactly what we're trying to do now. And actually, at the end of this, then soliciting, hopefully, people to participate in a clinical trial with us, ultimately. So Qui-Gon Therapeutics, so acute bleeding, leading cause of death in people ages 1 to 46. There are no FDA-approved pharmaceutical therapies out there. We don't take this lightly. Myself, my team came from Bayer. I'm the former head of the U.S. Innovation Center for them. I was their vice president, head of biologics research for them in the U.S. My team and I have over 100 years of collective industry experience. We're based in Berkeley, California, though I am living in Hanoi right now. Long story that we won't go over today, but yeah. Our lead asset, preclinical asset, is CT001, and we licensed this from Bayer in 2019. And I'll go over with you just really briefly who we are. I gave my introduction. I've got over 26 years of industry experience. I hired the individual who was doing the research projects within Bayer, hired him out of there. He's my chief scientific officer, Derek Sim. And then my chief medical officer is a surgical intensivist, has 20 plus years of experience, having done trials with Novo Nordisk and Eli Lilly as well on the sepsis side. So again, I think we're well qualified to go in this direction. The real challenges behind acute bleeding is, as you're well aware, it's unpredictable. In contrast to many other diseases where you get a diagnosis and then you set up an appointment, it's not like that here. It's unpredictable. And in fact, if you had rapid intervention, it's estimated that you would save 20 to 40% of the lives that are now lost. And the challenge here is compounded by the distance between the event occurrence, and that can happen in the home, that can happen on the battlefield, and where the resources really lie. And that is the staff and specialized equipment to actually deal with it. And consequently, why you don't see a lot of drugs out there, you see none approved, is that this is difficult to run a well-controlled clinical trial. They're high cost, high risk, and so it hasn't garnered the investment from the pharmaceutical and venture capital groups that we would all like to see. So the unmet need is truly something easy to administer, stable, and safe to enable rapid deployment, that is in the ambulance and on the battlefield, with healthcare workers closest to the event, the EMS, the corpsmen, those types of individuals who are going to get there first, right? And with that in mind, and in looking at the field, so I was in Bayer, and in taking up this task of trying to come up with something in acute bleeding, we asked the question, well, what is being used off-label, and can we solve for the problem there? So one of the things that has been used off-label still is factor VII. So this is a validated approach to address bleeding. It's FDA-approved drug for the treatment of hemophilia A, B patients with inhibitors. Off-label efficacy has been demonstrated in acute bleeding, but those are almost Hail Mary situations where it's a last resort, right? So it's you hear the stories of success and failure. The other feature that it has is that because of the work in hemophilia, it has a stable formulation. There is a lyophilized form, no need for specialty cold chain transport or storage, and it is IV administered, so no special equipment or training is required. The challenge, however, and why it hasn't really gone on is the safety risk. So it has a long, relatively long circulating half-life. And with that is associated thromboembolic risk for which it has a black box warning. So what we did was to solve for this problem, we made a fast clearing and enhanced potency molecule. So to improve the safety, we actually desilate this molecule. We actually take off the terminal sialic acid off the glycoprotein. And in doing so, you expose galactose residues. And those are seen by the natural recycling center in your body, the liver, by the acellular glycoprotein receptor. And so you have actual active clearance of the molecule from circulation. So we reduce the half-life from approximately 2 to 3 hours down to 3 minutes. So to compensate for that, we improved the potency. We took the GLA domain, the piece of the coagulation factor that's responsible for homing to the site of injury, and we modified it. And in doing so, enhanced the targeting and potency of the molecule, depending upon the assay, 2 to 5-fold. We still retain the high stability, that is the ability to lyophilize it, and the ease of use. It still is something we intend to go forward with IV administration. And so we feel we've made a safer and more efficacious next generation factor 7. And this is just a cartoon to illustrate that, moving from left to right. Intravenous administration, the molecule traverses the blood stream until it finds the site of injury. There it sets up shop because of the GLA domain that we've modified to improve the targeting and potency. But I think the most important aspect is the safety piece. That is, we desilate it for the short half-life. That is, this molecule is in essence a 1, maybe 2-pass drug, and then it is gone. And we've published 4 different papers on that. You see them in the lower right-hand corner. We've published 4 papers on this in peer-reviewed publications. And now we've finished our preclinical work and are working on manufacturing and going forward to the FDA. I want to give you a snapshot of some of the work that we did from those publications. I'm going to show you 3 panels that kind of outline the safety and efficacy of this molecule. So starting on the left-hand side, this is the PK model. And what we have listed here in red is CT001, and in blue is wild-type factor 7. That is actually Novo7. We actually purchased the drug. We list it in the publication as wild-type, but it is actually Novo7. So we really have the head-to-head here. And you can see that when you administer our molecule, it is rapidly cleared. It clears in approximately 3 minutes, where Novo7 is in circulation for 2 hours. So we predicted that would translate into safety. So we did 2 different safety models. One is a soluble tissue factor thrombosis model, where you're actually inducing a hypercoagulable state. You then introduce in factor 7 in blue or CT001 in red. And what you see is that in the case of the blue and Novo7, you lose approximately 60% of those animals when you administer Novo7 in that setting. In contrast, when you administer CT001, you see it fall basically on the same line as saline, which is shown in black. So that rapid clearance does equate to safety. We also did that in a ferric chloride-induced carotid artery thrombosis model. We saw the same type of outcome. So the question then was, with such fast clearance, would we see efficacy? And in the final panel on the right is this model. This is a tail amputation model. This isn't a prophylaxis model. This is tail amputation, bleeding occurs, and by definition of the amount of blood that's lost in this model, it is an acute bleeding setting. And then we actually administer our drug, Novo7 or saline. And what you can see is that you see a statistically significant difference in efficacy of our molecule. That is, the translation of that superiority of efficacy is actually translating in vivo. So with that as a preclinical package, we're going forward then with this as a drug. And where are we going? And this goes to the discussion with you today. And that is, we chose as our entry indication severe postpartum hemorrhage. And the reasons for that, I think, are obvious. It is leading cause, as Annette pointed out, leading cause of maternal complications and death worldwide, maternal. We know the general features. There are definitions around it. We know the serious, we know the treatments, we know the deaths that occur. And when we did clinical advisory boards, 2 when I was in Bayer, 1 since in coagulant, what we got was unanimous support for this in this indication space because the the diagnosis of the event actually occurs in the hospital. We're not playing with a variable of time. We can administer the drug at the time of diagnosis. There are common algorithms out there that define these kinds of things. Another added advantage is the patient is generally young. So in the spectrum of acute bleeding, we will not have the complications of potentially an elderly male like myself having had an accident and, having some comorbidity that you didn't know messing up the signal that you're seeking in your drug trial. The other additional piece is it does not require specialized training or equipment, right? So we think that this actually is the ideal setting, and that is what we're going forward with. With that in mind, then we did a study with Dr. Andra James at Duke University and another group in South Korea, where we actually looked at peripartum samples just to validate to ourselves that the events that we were seeing in the animal model actually, as best we could without being in the clinic, translated to efficacy. We published that down in the right-hand corner, and what you see is that that increase in potency is reflected in these patient samples as well. So our development strategy is severe postpartum hemorrhage is our entry indication in a controlled setting. We have orphan drug designation now. And then our expansion would be into the indication of trauma, traumatic brain injury, intracranial hemorrhage, and also expansion into the setting. This is where the safety is so important. Ultimately, if we're going to make a difference in this space, we want to have it brought to the ambulance, battlefield, and in this setting here, to the midwives, so that this could be administered by them as well, having proven it's safe and efficacious in a well-controlled clinical trial. So in summary, very briefly, we're engineering biologics fit to purpose. I showed you in this brief slide deck our Factor VII program. Novo-7 has been plagued by its thromboembolic potential that has a black box warning because of that. We think we've solved that by coming up with a novel mechanism for the unique clearance of this molecule by desilating it. The severe postpartum hemorrhage indication leverages positive Novo-7 randomized severe postpartum hemorrhage trial signal and recent EMA approval. So Factor VII is approved in the EMA for postpartum hemorrhage. And we think that's great. We believe we have the better molecule, the next generation molecule. So we see that as very positive. We have secured orphan drug designation here in the States. We have another complete, again, if you're the first one in the pool from a company perspective, we're not just looking at CT001 as a one-trick pony. We also have additional programs where we're going after activated protein C as also a treatment for acute bleeds. We've identified lead molecules for trauma and sepsis in that space as well. And really what we're looking for now, not part of this discussion per se, is that we're out raising capital to complete process development for CT001 production, bringing that to the FDA for a pre-IND and then IND meeting. And we're beginning to, and the reason I do want to speak to you about this is we are going to begin to explore sites for clinical trial participation, and we hope to be able to initiate clinical trials in the next 18 to 24 months if all goes to plan. So with that, I want to thank you all for your attention and allowing me again to speak to a very well-informed group on what we're going to try and do. And I look forward to your questions and hopefully to working with some of you in the future. Thank you.
Thank you, Terry. That was really exciting. I know I've heard some of the content here before, and I think it's great to have a variety of approaches, right, for postpartum hemorrhage. We have for cardiovascular conditions, cancer therapies, right? There's all kinds of nuanced therapies, and then in obstetrics we have uterotonics, and then now maybe antifibrinolytics, right? Like that's application. So I think that's really great. I had a question or two, but again, seemingly don't want to take over anyone if I don't see any hands. Let me just make sure. One basic question I had was orphan drug designation. What does that mean? I'm not sure I understand what that means.
It's more for the business side of things. So if you get orphan drug designation, what it translates into on the business side is it protects your market for X amount of time. right? There are benefits in terms of your BLA when you eventually file for your drug approval. Um, there are rebates, as it were, that come back to the company. So from a business perspective, it's advantageous to get orphan drug designation. There are certain uh, business features that that's a plus to. Has nothing to do with the clinical side.
Yeah, no, I was thinking clinical and because I think of
Yeah.
therapeutic orphans, pregnancy and children, but okay, not related. And
Yeah.
then one other question I had was related to the tail model that you all had
Yeah.
and efficacy. I was curious how saline had similar results profile, if you had that slide, to than the wild type Novo 7.
Yeah.
And I was just curious if you could share a little bit more about that, or maybe I misinterpreted that.
Yeah, no. So what you would see is if you go into the publication down at that right-hand side, this is one dose. We did a dose response. And what you see is that at this dose, because we have a more potent drug, we see this difference. If you dose higher, you will see Novo 7 actually coming up to the level of efficacy similar to what we have. So it's not that Novo 7 doesn't work in this model. It is, I would have to show you the dose response to both and then point these things out. So this is just a simpler way to present it.
I see. Okay. Yeah, I didn't see the 1 milligram per kilogram. Is that what you're saying? That's the,
Yeah.
okay. Got it. Thanks. Any other
Other
questions from the
questions?
audience here? Comments?
Well,
Okay.
if not, there is my email address for those who, like me, may think of something later and then want to reach out. So please do. Happy to talk to you about this. Oh, Neil.
Yes, hi, thank you very much. I just wanted to ask if the woman with postpartum hemorrhage had compromised liver function, would that affect the clearance time?
Yeah, in theory it would. Yeah. So we would have to, and it's an important question as we write the clinical protocol, because we are dependent upon that for the rapid clearance. That is true. There are other cells in your body that will. We've done some experiments to find out how much how much of your liver could be lost. Now, the point is that if you think about the amount that we would be administering, it is pretty minuscule. So we believe, and we've done it in the mice already, where we've gotten down to little little liver function and still seen the clearance. Um, so we initially we would, if there were compromised liver function, that would then exclude that patient in the initial trial for sure.
Thank you.
Sure.
I guess, Terry, just to comment on, are you looking for trials in the clinical world, or your clinical study in high resource, low resource, or any kind of regional country setting? I'm just curious because they would be
So
harder to make everyone included, but I think obviously it applies to everyone. So
yeah, so our initial approach was to focus in the US, to do it there first. However, talking to the Gates Foundation, they're like, well, could you do it in other countries? And I'm like, well, certainly you could. There, you just have to be thoughtful in terms of making sure you really had the infrastructure. This is not going to be a simple trial, right? You have the complication of the interaction of all the different moving pieces of the OBGYN, the anesthesiologist, the blood bank. You just start to put all those pieces together. It's not simple. And you would want to have the, clinical operations, and everything. There's just more complexity to it that's not there naturally that you would have to build in probably outside the US in some cases. But I don't think that excludes. It's got to be thoughtful.
yeah, thanks. And I guess one more question I'll ask for the folks on the call who manage as well women with bleeding disorders. Is there a specific role do you think that might apply to them, or equally as such for women without them? And we're talking about postpartum hemorrhage.
I'm sorry, could you go through the question again? I'm not sure I quite got it.
Sure, sure. And women with bleeding disorders,
Yeah.
I'm speaking broadly, not just one kind of bleeding disorder, but do you think that this potential therapy could be a
Hmm.
unique benefit for them in postpartum
Yeah.
hemorrhage setting, or it could, be more broadly applied to? So,
I think it could be more broadly applied. We do think about that. We have discussed the expansion even within women's health. I think those possibilities are there. Right now, we're just focused on postpartum hemorrhage.
great. Okay, well, thank you. Any other questions or thoughts? I appreciate um, the good dialogue and conversation from everyone. And then the last point I'll say is we'll announce or release the Unite Globe newsletter hopefully in the next week or so. And our next speakers will be in January, I believe. Sydney, correct me if I'm wrong. And then if you have suggestions for speakers, whether it's US or international based, always looking if there is a hemorrhage related theme with respect to postpartum hemorrhage, we would welcome speakers. So I appreciate if you know anyone to refer our way.